Hello, my name is Jordan Wolf, and this is my membrane concept map. So from the start, we had the plasma membrane, which is also known as the cell membrane, which is the biological cell membrane, and it's found in all cells, and it separates the internal cell from the external environment, and it regulates transports of substances entering and exiting the cell. And there's two subunits from that, and there's selective permeability, which is where the membrane allows the passage of molecules and ions to pass through and prevents others. And then we have the fluid mosaic model, which is by Singer and Nelson, and it's the structure of the plasma membrane. From that model, we have the phospholipid bilayer and the membrane proteins. First, for the phospholipid bilayer, that's a layer that's made up of two layers of lipid molecules, and it's a thin membrane that protects the cell. And it only contains small and uncharged particles, and will only let them enter. Then we have the Davison and Dinelli model. That's by Davison and Dinelli, and it's the model of the plasma membrane of the cell, but it describes the phospholipid bilayer, and that's between two cells of globular proteins, and how one faces the interior of the cell and one faces the exterior environment. From the phospholipid bilayer, we have hydrophilic head and hydrophobic tail. The hydrophilic head it faces outwards and it's polar, and then the hydrophobic tail faces inwards and it's nonpolar. From there, we have unsaturated and fatty acids and saturated fatty acids. Unsaturated, they can absorb more hydrogen, and saturated, they contain the max amount of hydrogen, and none more can enter. Going back up from hydrophobic tail, we have the proteoglycan, and that's inserted into the plasma membrane by a hydrophobic peptide domain. And then we go down and we have detergent molecules and apathetic molecules. The detergent, they're able to manipulate hydrophobic and hydrophilic interactions by disruption or formation of the cells. And then apathetic is the molecule that has a polar and a nonpolar side, and the polarity being the determining factor of how it's going to interact with other cells or molecules. Then from the model, we have the membrane proteins. Those are proteins that mediate the conduction of ions and molecules in and out of the cell. And there are three types. There's integral, peripheral, and anchor. Integral regulates permeability, and it's a gateway into the cell. And then peripheral, they temporarily attach to the plasma membrane but don't enter the core of the plasma membrane, which is hydrophobic. And then we have anchor membrane proteins, and those are located on the surface of the cell membrane and are covalently bonded to liquids within the plasma membrane. So from integral, we have enzyme membrane proteins, and those are small integral proteins, and they are hydrophobic, and they are located in the plasma membrane. Then we have monolayer, which interacts with the inner layer of the lipid bilayer. Then from peripheral, we have receptor membrane proteins, and those are in the phospholipid bilayer. They have a hydrophobic region as well. And then we have from anchor, we have lipid-linked membrane associated proteins, which are located on both sides of the plasma membrane, and they are covalently attached to lipids. And then proteins, attached membrane proteins, are attached to the biological cell membrane, the plasma membrane. From lipid links, we have glycolipids, which are on the outer surface of the lipid bilayer, and then we have glycoproteins, which are on the surface of the lipid bilayer. Then we go down, we have sterols and steroids, and those use lipid-derived hormones they're made up of lipid-derived hormones. So back up to membrane proteins, we have transporter membrane proteins, and those are for transporting substances across the biological plasma membrane, and they also form channels. Then from there, we have two types. We have active transport and passive transport. Active transport is the movement of particles in and out of the plasma membrane, and then passive transport is the movement from a high electrical chemical gradient to a low electrical chemical gradient. And there are three types of passive transport. There's a channel-mediated passive transport, diffusion, and osmosis. Channel-mediated being the moving down the gradient, but they don't require the assistance of other molecules to help push it through. Diffusion is carried out by the hydrogen-forming water molecules and solutes, and when the water molecules move in to surround the individual solute molecules. Then we have osmosis, which the molecules of the solvent pass through the semi-permeable membrane. And then from diffusion, we have hypotonic, hypertonic, and isotonic. Hypotonic, which is the solution outside the cell, is lower than inside the cell, so it is going to bloat because it's going to move inwards. And then hyper, it's the solution outside the cell has a higher concentration than inside, which is going to cause the cell to shrink. And then we have isotonic, which is the equilibrium. It's the same amount. And then from there we get the concentration gradient, which particles can flow up or down. And then we have the electrochemical gradient, which is for ions that can move through the membrane and down. And then we have membrane channels, which are a subunit of the electrochemical gradient. And those allow water and ions to pass through the electrochemical gradient. And then we have ion channels, which are pore-forming membrane that allow ions to pass through channel pores. And then we have ion-gated channels, which are pore-filling membrane proteins, and they allow a regulated flow of ions to travel in and out of the cell, or plasma membrane. I will show y'all